Okay, it's 1:15. Welcome, everybody. I wanted to let you know that I did post a couple Zoom recordings of chapter 4. So you should have a good chunk of that chapter already recorded in the course shell. I think there's one more recording that I need to do to wrap up chapter 4. That's on nucleic acids and protein synthesis. Some of you may have already started to look at that. If you haven't, you should. So I'll just open up the floor for questions related to whatever we've been talking about. Don't forget, tomorrow there's a lab quiz and the 30-point chemistry quiz as well. Since we have the chemistry tomorrow, are there any questions on chapter 2? Can you hear me?
Yeah.
Yes.
Okay, thank you. Everybody's good with the chemistry? Jess is asking about where she can find the study guides in the Blackboard course shell. Well, if you go to the course shell, Jess, and here we have the content listing, okay? And if you come down to study objectives by chapter, these are the ones that I told you I put together years ago. But nonetheless, I think they do a good job providing an overview of each chapter. So for example, here's chemistry, chapter 2, chemical bases of life, that we're having the quiz on tomorrow. And I've said repeatedly that I think that if you were to go through these objectives and answer them, in other words, develop a little study packet perhaps, that this would put you in good position, a good position for the quiz tomorrow. It's a very broad overview of the major topics. You'll have to get into more details, right? But those are the study objectives that I have mentioned in the past. And then I also posted here the student outlines, study outlines. This is from the actual author of the textbook, the book you're using for lecture. So you might want to check these out to see if those might be at all helpful to you. Does that help, Jess? Okay.
Sir, just related to the chemistry stuff, this is Ryan. I had a real quick question on, are you going to expect us to be able to draw like the detailed structure of each one of the types of atoms, or is that just is that too detailed?
Yeah, that's a good question, Ryan. I think that's too detailed. I'm not expecting you to do any drawing at all on the quiz or exam, whatever you want to call it. I think, you ought to be able to recognize, for example, a monosaccharide from an amino acid, from a triglyceride molecule, but it would be simply an identification sort of diagram if I put it on the quiz. But you do not have to draw out molecules or draw out atomic structures or anything like that. No.
Perfect. That answered my question. Thanks.
Sure. Anything in chapter 2 you'd like to review? Okay, how about the cell chapter? Anything on that? today or tomorrow in lab, we're going to be going over the cell cycle again. That, remember, involves interphase and the various stages of mitosis and so forth. So we're going to be hearing that again and looking at some prepared slides of different stages of the cell cycle. So that'll be kind of fun. We also introduced terms like diffusion, active transport, facilitated diffusion, osmosis, and so forth. We'll be examining that next lab tomorrow as well.
I have a question on the lab quiz. Is it going to be like on everything that we've done? Like, is it last lab, the lab before that, and this lab, or is it just going to be the lab that we're doing?
The quizzes are going to cover the material that we have studied since the preceding quiz. So if we go to the syllabus, let me just go to that real quick. Everybody see this getting pulled up? Yes?
We can see Blackboard, but not the actual document.
Okay. Now?
We're good now.
Yes.
Okay. So here's the lab schedule, and to answer your question, we had the first lab quiz last week over 1 and 2, but I've always said, and I'll say it again, that I reserve the right to ask a question or two about the exercises the day of the quiz, so I get a sense of whether you're looking ahead. So tomorrow, the second lab quiz is going to be over primarily the rat dissection and the microscope. You do not need to go back and worry about 1 and 2, because we're tested on that last Thursday. But you may also see a question or two related to exercises 5, 6, and 7 that we're doing tomorrow. That particular type of question would be a question that I would ask to assess whether you have looked ahead, you're familiar with what we're going to be doing in those exercises. So it again is sort of my way of gently prodding you to look over the exercises before you walk into lab so that you're not totally oblivious to what we're doing. I'm not saying you have to know everything there is to know about 5, 6, and 7. Okay, that's not what I'm saying. I'm saying you should look them over so you're fundamentally aware of what we're going to be doing in those exercises. If you don't read ahead, you will not get 10 out of 10 on the quiz. So that's your, you know, I'm just giving you fair warning, okay? About that. It's my mean way of making you look and read ahead. Does that answer that?
Yes, thank you.
Sure. Other questions on anything? Okay, there's some stuff in the chat. Let me look. So Skylar, did I answer your question about the quiz? Okay. Yeah, I had heard yesterday that the storm that came through, what day was it? My day's all mixed up. Monday, I think, knocked out power. There was a lot of wind and some trees fell. I know in Olean, Worth W. Smith Hardware down the street, I stopped by there the other day, they had no power. So I suspect that's what's going on, that there's been some power outages due to that windstorm, right? That came through, I think.
Yeah, and it affected us today. We just
Yeah.
lost power for a little bit. So I just wanted to make sure that you got us in because we got on here a few minutes before class started and then it shut off. So I just wanted to let you know that.
Okay, thank you.
Yeah. Last
Hopefully-
week I didn't have power for 4 days, and then this week it was 2 days. So hopefully it'll be back on soon.
And what are they telling you about why?
Well, last week it was because of all the trees were down. All my lines, because I live out in the woods, they weren't too worried about where I was because I was only one person. So I had to go 4 days without power. And then this time they had a couple trees down, and then also some transformers blew. So that's been causing a lot of power outages.
Yeah, I live in Eldred.
Yeah.
I'm at my parents' house in Bradford. I'm at my parents' house right now in Bradford, but I originally live in Eldred and my power was even out along with Bradford's for like a day or two. I had to get a generator for
Oh,
our house for a little bit.
huh. I didn't realize it was that widespread. I had heard up Erie County way they had lost 5,000. Some people had lost power early in the day, and then by the end of the day, there were like 1,000 plus people out. So interesting. So any questions about chapter 3 on cells you'd like to go over? Or we could even talk about chapter 4 if you got into a little bit of that. I'm open to whatever you'd like to review or talk about.
I did want to ask about chapter 4. I read those, or I watched those lectures that you had posted on for the PowerPoints and stuff. And on the last one, it said something about you uploading for 4.5. Were you uploading for that one, or are we just reading up on that one? I know we had to read all the chapters, or all the chapter 4, but
I just had not gotten to that yet. I ended the last one with a comment about we're going to stop here, and then the last lecture, if you will, of chapter 4 will cover 4.5. So there's nothing uploaded yet. I will get that put up. I would guess probably by this weekend, because if we look at the schedule, the lecture schedule, we have this week and I think part of next week to get through chapter 4. So I'm adhering to the schedule, the lecture schedule, and trying to keep up with that, or maybe just a little bit ahead even. Yeah, and chapter 4 covers quite a bit of information, and the chapter itself is not super long. I think it goes, well, let's see, 23. Yeah, I mean, it's only like 23 pages long, which is actually quite short for our A&P book chapter. But I do take some time and I go through and very careful analysis of some of these topics. So you definitely will want to get into chapter 4 and start looking at that. I cannot believe that you have no questions about anything.
Sir, I was going to say that explaining that diagram for cellular respiration, I mean, I think the hardest part of that is trying to understand. I mean, I guess the question is for you is how much detail, again, I know I asked this question 1,000 times, but like, what kind of level of detail are you wanting us to understand? Are you wanting us to understand of like what is produced? Like, I know you want us to understand like, hey, every time you go through the Krebs cycle, you have to go through twice for every glucose that you're using. Like that part I get and understand. Are you also wanting us to just understand like the products of each of each one of those the three things. So, you know, the Krebs cycle, glycolysis, and then the electron transport.
Okay, that's a good question. Let me pull up the PowerPoint. Okay, do you have that in front of you now?
Yes.
Okay, so for those of you that have not gotten into chapter 4 yet, this isn't going to probably make as much sense as it is to Ryan and those of you that have started to look at it. It's like my screen froze here. Okay, here we go. So his question relates to, come on. All right, let me try something else here.
While you're waiting for that, sir, I think, at least from my standpoint, I don't know what everybody else thinks, but I think the first three sections of chapter 4, 4.1, 4.2, 4.3, that stuff, you know, the basic structures of the reactions, things like that, those are easy to understand. It's the meat of the chapter seems to be chapter 4.4 and beyond that. That's the stuff that I know I'm probably going to have a lot of questions on.
Yeah, and that's what I would hope and expect you to say. So thank you. You're right. The first few sections are really not that hard, or shouldn't be. Oh, my computer is just like frozen here. I don't know why. I want to pull it up so I can talk about the sections, Ryan, so just hang on a second here. All right, let's see if this works. Okay, do you see the PowerPoint now?
Yes.
Okay, and is it scrolling through the slides now?
Yes.
Okay, thank you. So this is the segment coming up here in chapter 4. That you were asking, how much detail do we need to know about glycolysis? How much do we need to know about Krebs cycle? And the answer to that is you should know what the starting compounds are, what do you end with, and what do you make along the way. For example, in this diagram, you see the citric acid cycle. You do not need to know the names of all of these compounds that I'm circling. Okay, not important. You should know that citric acid is the compound that enters the mitochondrion and begins a whole host of reactions, as we can see evidenced by the blue arrows, each of them indicating a reaction, right? So you should know pyruvic acid, and I stressed that in the PowerPoint, you know, the narration that I did on Zoom. I did also mention this compound here, citric acid. And I guess I would know that because it's the first compound made in the actual cycle of reactions. So this first set of reactions are sort of what lead into the true cycle itself. So when we talk about citric acid cycle, we're talking about this circular cycle of reactions. This sort of leads into that. And the reason that I think it's important to know citric acid is simply because that's where it gets the name citric acid cycle. It's the first molecule made. So that's pretty easy, right? I also said, if you watch this video that that I did the other day, that these gray circles represent what?
Carbon.
Right, and it says right up here, carbon atom. So this is a this is a 6-carbon compound, which results from a 2-carbon compound combining with a 4-carbon compound. Right, 2 plus 4 is 6. So when we talk about pyruvic acid coming into the cycle, we pull a carbon off of that, and you can see what that forms, CO2, which I talk about being a waste product. That's what you're exhaling right now as we speak, right? You know your cells make CO2. So that's where some of it gets made, right here at this particular reaction. And then this 2-carbon compound is what basically feeds into the cycle and combines with the 4-carbon compound that was made at the end of the preceding cycle, right? So what ends up then happening is you take this 6-carbon compound and you basically break it down back into the 4-carbon. Well, that means you've lost 2 carbons, right? 6 minus 4 is 2. And where are those? Well, here's one and here's one. Do I need to know where they're made here in the cycle? No, I just should understand the concept that I'm making a couple carbon dioxides here in the citric acid cycle or Krebs cycle, and I made one leading up to it here. What else is made from top through the whole cycle? You should know that. In addition to those 3 CO2s, what else does the cell generate? And I had a slide about that on the lecture too. Well, we make some NADH. We make one here, and then we make 3 in the actual cycle. We also make an FADH2, and I talk about the function of those particular molecules. They're electron carriers. And we also make a single ATP, right? So to answer your question, what I would expect you to know is A, where does this occur in the cell? And we talked about that here, as well as in the recording that I did. So you need to know, where does this cycle happen? The answer is in the matrix of the mitochondrion, in the very core of the powerhouse organelle. And it starts with a single pyruvic acid molecule coming in, which is produced at the end of glycolysis. You make 2 of those. And in so doing, the cell generates 3 CO2s, 1, 2, 3, 4 NADHs, 3 really produced in the cycle, 1 made early on in the reactions leading into the cycle, 1 FADH2, and 1 ATP. So to answer your question, that would be the detail I would expect you to know with regard to the citric acid cycle. You do not need to know the names of these intermediates other than maybe citric acid, because I talked about that a little bit. You should know pyruvic acid as well. That's an important compound, but that was made at the end of glycolysis. So we can go back to glycolysis here and you can ask the same thing. Well, do I need to know the names of these intermediate molecules? No, you don't. Do I need to know what I start with? Yes. Do I need to know what I end with? Yes. Do I need to know what I make along the way? Yes. What do I make from top to bottom here? You tell me. Basic byproducts of glycolysis. What would you list?
Well, we produce 6 ATP. Looks like.
No.
Or wait, no, 4 ATP, right?
Look close. There's 4 produced toward the tail end, right? But what's going on up here?
That's actually burning it off. So we actually
Right.
producing 2 then, two net. I guess.
Right. That's exactly right, Ryan. You come out ahead 2. Yep. So there's 2 ATPs. Okay. What else do we make?
Well, we make NADH. It looks like there's 4 of those.
No.
No, wait.
Right here.
2.
2 of those. Yep. And that's basically it. Other than the 2 pyruvic acids, of course. Don't forget about those.
Right.
Yeah. But
So
That's
if I understood, like for example, like with the previous slide where we were going over the Krebs cycle,
Mhm.
if you understood like okay that one pyruvic acid produces 1 ATP, 3 carbon dioxide, 3 NADH, and 1 FADH. If you understood that, then that's the basics of what you really want us to get, right?
Yes, but I would just correct you about the 3 NADHs. You're talking about these here in the cycle. Don't forget there's one made up here.
Oh, that's right. That's 4. Okay.
Yeah. Now I did say in the recording that I did, I said we can just lump those all together and call it citric acid cycle 4 if you want to do that. But the reality is that the technical reality is there are 3 made in the cycle and there's 1 made prior to the cycle. But I don't want to muddy the waters any more than I already am. So if you want to just include this as part of the whole thing, I'm okay with that.
Perfect.
Yeah.
So basically, if we just wrote ourselves like down, okay, so the Krebs cycle obviously is taking place, like you said, taking place in the matrix of the mitochondria. You understand that, and then you understand, okay, pyruvic acid, when you take it through the Krebs cycle, you get ATP, 3 CO2, 4 NADH, and 1 FADH2. And if you got that, that's
good.
basically the understanding of what you really want us to get, right?
Yes.
Okay. And if I did that for every single one of these steps, you'd have that for the electron transport section and then the glycolysis section. If you understand all those things, basically you make yourself a little chemical equation so that you remember, hey, this is what's in here. That's really what you want us to have, right?
Right. Somewhere in my PowerPoint, I think it was here, I give you that.
Right. Okay.
Yep. I summarized this for you, but you should understand where I'm getting these numbers and these coefficients from. Yeah.
Perfect.
Yeah. Now, the electron transport chain is not as tidy. It's more understanding the mechanism, right? The flow of electrons from protein to protein to protein within the inner mitochondrial membrane.
Right.
Yeah. And that on average, for each NADH that liberates or gives up its electrons, the cell is going to generate approximately 3 ATP molecules, as opposed to 2 for each of the FADH2 coenzymes.
Got it.
Yeah. And I'll tell you, just to be perfectly honest with you, this stuff is not easy. If you've ever studied this, it taxes the brain cells because there's a lot of chemistry, relatively, and some of this is something you have to kind of visualize. It's not like you can take the ulna and measure its length. You know, this is a theoretical, not theoretical, but it's more of an abstract set of processes. It's not made up. This is true chemistry. Okay, people won Nobel Prizes in chemistry for understanding this. This is not theoretical. I shouldn't use that word because it's been discovered to be the way it is. But it is. I think it's fair to say most students have a hell of a time with it because it's very technical. And sometimes it's like I can't possibly understand this, and you have to go over it a couple of times. You got to ask questions about this. You got to synthesize it. You can't just look at it once and just know it. This is going to take a little bit of stretching of the neurons. And the reality probably is also that when you take A&P from other people, that they don't go into as much detail as I do. And that you can argue either way, right or wrong for that. I happen to think it's kind of important. And I'm just scratching the surface. I'm not getting into a lot of the technicalities here. The reason that I go over this in this detail is because a lot of you are taking micro after A&P, right? I'm going to assume that you know the basics of these processes because we utilize them in microbiology lecture. So you're not done with it. You're going to hear it again. And I just think it's good to hear it here in A&P. This is why, again, students should have some basic knowledge of chemistry if they're going to understand biology. You can't separate the two. They go hand in hand, whether you like it or not. It's the way it is. That's why we have the chemistry prereq for this course. How about other questions on chapter 4, if you have any, or whatever? Because I think a week from Thursday is our first lecture exam. So that's coming up quick. We'll talk more about that, early next week, but 80 points over chapters 1, 3, and 4. We will not put any of chapter 2 on the lecture exam. That, of course, is coming up this week. That 30-point chapter 2 chemistry exam. If you don't want to ask me verbally, write it in the chat, the question you might have. Some people are just afraid to talk, I think. Okay, well, I'm fine with concluding class if nobody has any questions. This is the approach we adopted early, was that I was going to post this stuff, you were going to look it over, and then you were going to ask questions and get clarification. So that's the premise I'm working under, and you guys got to ask.
What papers are we supposed to bring to lab tomorrow?
Say that one more time, Victoria.
What are we supposed to bring to lab? Like, what are we supposed to print out to bring to lab tomorrow?
Well, we've said that if you go to the lab schedule, right, and see what we're doing each week. And so, do you have the syllabus there?
Yes, I do.
All right. Let me pull it up. It says 5, 6, and 7, doesn't it? Plus the lab quiz and the chemistry exam. If we go to the stuff to copy and bring to lab each week folder, I'm going to pull that up right now. I'm going to share that with you. Okay, everybody see that? It lists the exercises in chronological order, time-wise. For this week in lab, we're doing exercise 5, 6, and 7. So you see here it says SAMSO cell model that you need to bring. That's the handout for exercise 5. There's nothing for 6 or 7, obviously. But when we do exercises 8, 9, and 10 and beyond, you can see there are some handouts to bring, depending upon what the exercise is. These I'm not bringing for you, right? You're going to copy this off and bring this with you. So the stuff there in black, I have freed up the keys as well as the fill-in parts of the models. So you don't have to bring the key if you don't want to. I'll try to remember to bring hard copies of that each week, but certainly you should bring those diagrams that have you fill in the number that corresponds to a particular structure. Yeah, that's what those models pretty much are. So I have said that it would be helpful maybe to try to print all this stuff off And then you can use the 3-hole punch if you'd like in the lab or to make that, and then throw it into a small little cheap binder, and then bring the binder each week, and then you've got everything you need. You don't have to worry about, well, what do I need to bring this Thursday? If you do that, I can promise you that A, you'll either forget, B, you'll think of it when you walk in and you won't have it with you. You'll be rushing in the back room to try to print it off, and the printer will be out of paper or something. I can guarantee you it's not a good thing to do. Okay? Don't procrastinate. Get it done ahead of time. Suck it up. Do all the printing. Bring the packet with you, the binder. Then you're done. You've got everything you need for the rest of the course. That's my recommendation to do. Because I'm not going to be providing you again with those handouts. So you got to get in here, see what's going on, and bring what you need to bring. Is everybody clear on that? Haley, are you clear on that? These handouts are in the Blackboard course shell.
Sir, just related to the quiz, the lab quiz again, you said it's about the rat dissection diagram in the microscope. So I guess I'm trying to think of like some questions and some of the things you might ask on that. Like, I mean, I suppose you could ask about, okay, so you have the objective, right? And then you have the lens and or I can't remember what it's called, the top portion of the microscope. You could ask a question about, well, what's the magnification if you have an objective lens that's this and this, and you can kind of go off of that. But I was gonna just ask in like kind of general questions of like, what are you looking for as far as that's concerned? Because the rat dissection diagram, I mean, we were just basically taking a rat apart at that point, understanding how to do that. All that.
Well, in terms of the microscope exercise, which I think was it 4? I think it was 4. We went over the parts of the microscope, right? From the top to the bottom, literally. And we described all of the structures and what they did. So that's all fair game. I could put a microscope up in front of the class and put some pieces of tape on it that are numbered and ask you to identify the part of the scope, number 1 or 2 or 3 or whatever. So it's helpful, I think, to go back and review the different knobs and what they do. In addition to, talking about how you can calculate the total magnification by multiplying the ocular by the objective, right, as you mentioned. So I didn't go over that because I just like to talk. I did that because you need to know the different names of the different parts. And then, of course, we talked about how to go from scanning to low and low to high, right? We talked about working distance. We talked about the field of view. We talked about how to regulate the left ocular diopter ring. Remember that? All that stuff, fair game. So it wouldn't be a bad idea to take notes if if you feel the need to take notes, because that's fair game. And then as far as the dissection goes, yes, we of course cut into the animal, but we also talked about different organelles and what they did, right? Some of them, not every organelle, or every organ, I should say. But we talked about quite a few of the organs, and we found them. But if I'm talking about the role of the ureters, let's say, as it conveys urine from the kidney to the bladder, even though you might not have seen it, if I talk about it in terms of where it's located, fair game. I don't know, does that help clarify that?
Yes.
Okay. If you don't have your lab books, I would say, why don't you have your lab books? We're in the 3rd week now. Courtney, are you here? Courtney. Okay, any other comments, questions? We'll do the lab quiz at the beginning. Yep. And then we'll do the chemistry quiz toward the end of the lab. Yeah, that's kind of my thinking. So Courtney, you say you ordered 3 weeks ago and it hasn't come yet. I think I would do some phone calls and see what the heck's going on there. Maybe you could borrow somebody's book and make some copies. Any other questions? Going once, going twice. Okay, I will call an end to our class, I guess, then. I'll stay here in Zoom for the next, half hour or so before my micro lab starts. So if anybody wants to hang around, ask questions, you're certainly welcome to do that. Otherwise, I'll see you guys in lab tomorrow.
Now, you said that we need exercise 6 and 7 for tomorrow. Is that just in our lab manuals? Is that right?
We're doing 5, 6, and 7 tomorrow, and that refers to the lab manual exercises, right?
Okay, sounds good. And then just print out 5 for tomorrow as well
Yep. That's right.
from the what we need to bring to class. Okay.
From Blackboard.